All right, guys, let's talk about enzymes. Second part of Chapter 8. All right, enzymes are catalysts, and in chemistry, a catalyst is defined as a substance that makes a chemical reaction go faster. In living organisms, it basically is enzymes are factors that allow chemical reactions to happen altogether. Without enzymes, most of the reactions in the body wouldn't even happen. And most reactions do require some kind of enzyme. Catalysts are not consumed by the reaction. In other words, the enzyme is still intact afterwards and can do it again, which is kind of a nice feature. So they do not get broken down by the reaction itself. Enzymes are inevitably proteins. Proteins are the only biological molecule complex enough to be able to function as an enzyme. Because enzymes require very complex three-dimensional shapes, and proteins are the only ones that can provide this. Even DNA with its complexity of the different letters that it has with the bases, does not form anything complicated that would work for this. RNA can come close. It can fake it a little bit. It can act like an enzyme. Because it can form three-dimensional structures, but nothing compared to proteins. Proteins are much better at that. All right, let's take a look at an example. Let's take a look at the hydrolysis of sucrose, in other words, splitting sucrose into glucose and fructose. That is accomplished in digestion by the enzyme sucrase. Sucrase makes this reaction happen. Without the sucrase, this would take very, very long, and the enzyme makes it more efficient. Enzymes tend to end with ACE. Their names tend to end with ACE, and often their name implies something about their function because this works on sucrose. It's called sucrase. Not real creative, but makes it easier to deal with. And obviously, you guys remember these structures. You remember the structure of glucose. You don't have to know fructose for the test, obviously, but maybe you remember that sucrose is made out of glucose and fructose linked together with this glycosidic linkage right here. All right, so enzymes basically have this function of working on and modifying the activation energy of a reaction. So chemical reactions, and especially ones in living organisms, tend to have a pretty steep activation energy. That's the energy you have to put into a reaction to get it to then occur spontaneously. You have to put a little something in, an investment, and then you can have the reaction proceed and maybe extract energy out of it. Activation energy. All right, so the enzyme is going to reduce this for us. It's going to help us get over the hump more easily. You can think of the activation energy as the maybe the heat you have to put in to get a piece of wood like a log to burn. You have to take a match or use a lighter and put a bunch of heat into it before it'll actually start burning. Another example would be if you're going sledding and there's a little hump that you have to get over, a little hill of snow at the top of the hill that you get over. You have to push your sled over this hill, and then you can go down the hill spontaneously, which would then release energy from that reaction. All right, so what the enzyme is going to do is it's going to reduce the activation energy. This would be the activation energy with enzyme present, which is lower, and this would be the activation energy without the enzyme, which is quite a bit more steep. In reality, this would probably be a lot lower. So with enzyme, it would be down here, and without enzyme, it would be up here like this. So this diagram is a little bit unrealistic in that this is still pretty steep. In reality, this would be like way down here. So it's very easy to get that reaction going and get it to that downhill part where the energy can be released. All right. Keep in mind, based on what we learned earlier with exergonic and endergonic reaction, this would be exergonic. And the reason why is because it has a negative delta G. Negative delta G means it's spontaneous. We start at a higher energy and we go to a lower energy. It's a downhill reaction. All right, so enzymes have different parts to them. You may know that they have an active site. This would be the active site. Substrate will bind, that's the molecule that the enzyme acts on. All right, this will bind here at the active site. It forms the, I don't know why my pen is so sluggish. Sometimes it does that. It's very annoying. It forms the enzyme substrate complex. And frequently during this, induced fit will happen. An induced fit is just simply the enzyme biting down on the substrate and clamping down on it a little bit to hold it tight. Many times then the substrate will get broken down. So if it's a catabolic enzyme, it's going to break it down, and then those products would be released. So we then have the pieces here that then get spit out of that active site. Okay, those would be the products. All right, the enzyme is still intact. It can do it again. It can do it as long as there's substrate available. The enzyme does not get broken down or consumed in this process. This picture just is a realistic computer-generated three-dimensional space-filling diagram of an enzyme of what it would look like. The color is obviously not real. It wouldn't have little bubbles like that, but this would be the overall shape of the enzyme like this. The active site looks kind of like a mouth, and it looks like the mouth is open here. And then once the substrate binds into that active site, the enzyme clamps down on it like this, and the mouth closes and sort of bites down to hold that substrate tight to do the job. How do enzymes accomplish any of the things they accomplish? Well, they have a number of different strategies or mechanisms that they engage in. The two easy ones to understand are these first two. They're the little like real logical ones. Orienting the substrates correctly. For example, if you have something like this, maybe you have your first molecule that looks like this. You have your, my goodness, you have your second molecule that maybe looks like this. They have to be lined up just right to fit into each other like this. If this was reversed and the other one was facing this way, for example, it wouldn't fit into this opening. So the enzyme will align the two molecules that have to be joined together and then bring them very, very close together to allow those bonds to form that have to form in an anabolic reaction where you make a larger molecule. A second thing that might happen is that if it's a catabolic reaction where you're breaking something down, so let's say this is our molecule that has to get broken, the enzyme may start to sort of bend the molecule and strain the bond that's in the middle there that has to get broken. Okay, and eventually it's going to snap right here at this point. Okay, so then when it's done, you got two pieces. So by straining the substrate bond, like if you took a pencil and bent it in such a way that it was strained, and it would be very, very easy to break then at that point. It just takes a little bit extra effort to break. All right, so that's a second possibility. A third one would be providing a favorable microenvironment. What that's all about is providing for something like maybe the correct pH or the correct non-polar R groups that have to be present to make it a sort of a hydrophobic environment, maybe a non-polar environment, or maybe a hydrophilic environment to have those kinds of like polar or charged R groups in the middle. Enzymes can do that. If you take a look at our little monster guy here, this cave that forms here, this sort of chamber that forms is secluded away from the outside world, and it forms a microenvironment right in here excuse me, for that substrate to have optimal conditions to have the changes happen. And then finally, enzymes might, at the active site, bind covalently to the substrate to get a really strong sort of a grip on it to be able to manipulate it into what has to happen to it. And that covalent bond then has to be broken when it releases the substrate. All right, we know that enzymes have optimal conditions, and those could be, relating to pH or temperature and things like that, salt concentration. we can see in these graphs that these activity rate graphs, we can see that for temperature on the first one, clearly this enzyme that's shown here in black has an optimal temperature at about, if you drop this down, maybe something in the neighborhood of 37 degrees. That happens to be body temperature, and this is a human enzyme. So some kind of an enzyme that's in a human being works best at 37, which makes sense because that's human body temperature. We're talking about Celsius right now, 98.6 Fahrenheit. Any colder than that, an enzyme activity will slow down, so the activity goes down, the rate of the reaction decreases, because molecular interactions between substrate and active site would decrease, and the amount of sort of joinings between the two would be reduced. If you increase the temperature even slightly, you go too high, the activity goes down, because at that point the enzyme is going to get denatured. All right, so denaturation happens at that point where the enzyme gets usually irreversibly broken. The reason why that happens is because molecular interactions are so fast at that point that the enzyme three-dimensional structure breaks apart. Water molecules are bumping into the enzyme at high velocity, almost acting like little bullets, and causing damage to the three-dimensional structure of the enzyme. So like in a place where there's hydrogen bonds or weaker kind of interactions in the tertiary structure, the enzyme may bend and shift its shape in such a way that it cannot come back and recover from that. A good example of that is a egg getting dropped into a hot frying pan. The protein of the yolk and the albumin will denature and change into a fried egg. And even if you cool it down, it cannot go back. Here's a thermophilic enzyme. This is from bacteria that live in hot conditions. That's normal for them. So their optimal temperature is maybe 78 or something like that, or 76 degrees or whatever it might be, Celsius, a much hotter kind of environment, because that's what they're used to and that's where their enzymes work best. Any hotter though, and they also go down, and any cooler, their enzymes will not work because of reduced interactions. pH is a second thing that is very important for enzymes to function. This enzyme over here is called pepsin. It is found in the stomach in the human or in mammals. And in the stomach, we know the pH is very low. There's hydrochloric acid to help digestion and actually to help the pepsin work. Pepsin works best at pH 2. Okay, so that's another reason why the pH is so low, because pepsin actually requires a low pH to become active. Any lower than that, there's too many hydrogen ions now in this acidic condition, which then interfere with ionic bonds in the tertiary structure, or hydrogen bonds can get broken up by these hydrogen ions and then cause the protein to denature. At a higher pH, we don't have enough hydrogen ions, or we have too many hydroxide ions, and we get denaturation at that point. So the whole idea of, on the left side of the graph up here, we don't get denatured, that's no longer true here. If you go acidic, you're going to ruin the protein. You're going to ruin the enzyme. That good example of that would be like milk that's exposed to lemon juice or vinegar will curdle, and even if you raise the pH up again, it doesn't come back. It's ruined for good. It's irreversible. This is trypsin, which is an enzyme that's found in the small intestine that likes normal, like neutral pH. So we see that it works best at about 8, or it's even pretty good at 7. So the pH of the small intestines is above 7, but real close to neutral, maybe up to 8. And trypsin works best then. Any lower than that, it doesn't work. Any higher than that, and it stops working because it gets denatured. All right, so that deals with the concept of optimal conditions for enzymes. There's other conditions we could talk about, like salt concentration, but temperature and pH are really the most important to understand. All right, some enzymes require cofactors. They require helper molecules. Those cofactors will allow the enzyme to work. This is also a way to be able to turn the enzyme on and off. Cofactors could be inorganic or organic. Metal ions, for example, could be a cofactor. Vitamins, things like that. If they're organic, they're called coenzymes. So a lot of times vitamins are coenzymes that help enzymes work better. Sorry about that. All right, enzymes can be inhibited by outside factors like poisons or substances that could harm us. These inhibitors of enzymes could be competitive, or they could be non-competitive. If they're competitive, they bind the active site. They bind this part right here. Okay, so if you get something like this coming in here, oopsie. Okay, it's not letting me draw because it hates me. All right. Oh my goodness. Please let it work. Okay, so that might be an inhibitor that binds at the active site right here, and the substrate comes in and it can't bind because this is blocked. A non-competitive inhibitor would bind somewhere else. So if you have your enzyme like this, maybe the non-competitive inhibitor binds right here. If it binds right here, it might have an effect on the active site and close the active site up. Notice the mouth isn't open because this non-competitive inhibitor was binding over here. This would be the competitive inhibitor. Toxins, poisons, pesticides, antibiotics are all examples of these kinds of inhibitors. All right, so this is kind of a more elegant drawing of what I just showed you. Normally the substrate would bind here at the active site, but now this competitive inhibitor bound, and it's preventing the substrate from binding, so the substrate can't bind. That's competitive because it's at the active site. Here's a non-competitive inhibitor somewhere else at a distal place, changing the shape of the active site, so this doesn't work anymore. So notice how this over here sticks up like this normally, but now it's bent. So now the substrate is unsuccessful in binding correctly, and maybe it falls off too easily. It doesn't get clamped in correctly to stay and have the effect happen. Sometimes natural processes in cells will regulate enzymes to be able to turn them on and off. That regulation is called allosteric regulation. So allosteric regulation is not a situation where it's a poison or some substance from the outside that does harm, but it's actually a natural process that is supposed to happen to allow a cell to turn on or off, turn that enzyme on or off. Okay, and usually that regulation happens at a distal site. So let's take a look at it. Here we have one picture of an allosteric inhibitor and one picture of an allosteric activator. All right, so here we have, if we take a look at the left, we have the enzyme. We have the active site right here. We know that's the active site, by the way, because this is the substrate right here. Okay, so when the allosteric molecule is not bound, notice the substrate cannot bind correctly. It doesn't fit. But when the allosteric substance is bound, then all of a sudden now the substrate can bind. Now the substrate fits in here and it is working correctly. That tells you that this yellow molecule must be activator or inhibitor. If you said activator, you were correct. It activated the enzyme and allowed it to work. Without it, it doesn't work. All right, this substance over here is not bound, so this molecule is not bound, and the substrate is happily bound. Everybody's great, and it's working correctly. But when it does bind, when the purple one binds, the substrate gets kicked out because the shape changed over here of the active site, and the substrate doesn't fit anymore. So this must be activator or inhibitor. Well, the only one that's left is an inhibitor. So we know that this one must be the inhibitor, and this one must be the activator. So just by looking at the picture without labels, by looking at what's happening in the sequence of events, we can figure this out. Wouldn't that make a good test question? Yes, it would. All right, a specific example of this kind of allosteric activity is an enzyme or a group of enzymes in many living organisms have these that are called caspases. Caspases are enzymes that are responsible for apoptosis. You might have heard of this before, or maybe not. Apoptosis is cell death or cell suicide. And cells kill themselves. Suicide. Cell suicide. Sometimes cells have to die for the good of the organism. If they're extra, or if the cells are actually causing problems, or if they're cancer cells or something like that, it's actually a good thing if the cells die, and the caspases make that happen. Okay, so here we have a situation, we have a caspase enzyme that oscillates between the active and inactive form. Okay, so in the active form, it's able to bind the substrate and it's able to cause its effect, which then causes cell death. That's going to lead to cell death up here, which sometimes is appropriate and sometimes is actually going to be beneficial. But it goes back and forth between this under the normal circumstance, and while it's doing that, while it's for the short time that it's in the active form, it can bind the substrate and then take care of its job. Sometimes what happens though is a molecule over here binds, an inhibitor binds, and locks it into place, locks it into the inactive form. So now it's inactive. And it actually forms a disulfide bridge. It actually like covalently bonds to it, keeping it inactive. And you might want to, you might be wondering, why a disulfide bridge? Why so extreme? Because normal cells, our cells that are doing their thing in our body, that are doing their job, those cells have these enzymes in them, but in an inactive form. So the enzyme is being prevented from killing the cell. So it's not actually going to kill the cell under normal circumstances, but if this was released somehow, the cell would die. This enzyme would become active and then cause the cell to die. So that actually is a very dangerous kind of a setup, because we have these like killer proteins in our enzymes, in our cells, that could kill cells and then eventually kill the whole body if they were activated. But these inhibitors prevent that from happening. It's only when it's time for the cell to die, that's when these inhibitors let go, and then it goes into this mode and then causes cell death. All right, so that's apoptosis, and PowerPoint knows how to spell apoptosis. That's impressive. What if I say it differently? Apoptosis. Oh, it knows how to say that too. Okay. Lots of ways to say that word. My son has informed me that it's pronounced apoptosis, so I'm going to try to say it correctly to make him proud. All right. All right. Sometimes, oh, this is actually just more. Oh yeah, sometimes enzymes are actually made out of many subunits. They have quaternary structure. So this would be like 4 tertiary structures bound together. And if you just need one activator to activate them all at once. So sometimes that happens. Or one inhibitor can lock them all in the inactive form. This just shows, I guess, that some enzymes are not just one molecule, but they're these clusters or complexes in quaternary structure, multiple proteins stuck together, subunits, I guess you'd call them. Cooperativity is another thing that can sometimes happen, and that's when a substrate actually binds, not an inhibitor or an activator, but the enzyme is normally in inactive form, but it can oscillate back and forth between the different forms. And once substrate binds right here, it locks all the rest of the active sites into the active position. So what it does is it takes all the other subunits and turns them on also, and increases the affinity or attraction to the substrate, so that now the other substrates are going to bind more easily here at this point. So that's cooperativity, because this is cooperating with the other sites and turning them on, getting them active once some substrate binds. This is a good thing if there's a lot of substrate. We don't want it to take too long for the substrate to bind. We want to make it more attractive and efficient to get that substrate taken care of. All right, one of the last things we're going to talk about is feedback inhibition, and this looks actually really complicated, but it isn't. So just give it a chance here. Don't get too intimidated by this next picture. It's not so bad. So we're going to talk about an enzyme called 309 Deaminase. This is just simply an enzyme that's responsible for the synthesis of amino acids, and the product in this reaction is isoleucine. So let's take a look. So the product at the very end is isoleucine. That's what we want. That's what we want to make. We start with the original substrate way in the beginning of this cascade, in the beginning of this chain of reactions, is 309. PowerPoint doesn't like me saying 309. 309. It doesn't recognize O. 309. Oh my goodness. 309. No, it doesn't. O. O. It doesn't hear me say O. Oh. Wow, that's so weird. It knows all these complicated science terms, but it can't even do O. That's very strange. Okay, sorry, I'm wasting time. Okay, so what we have is this chain of events is not really all that important, but one of the enzymes in that chain is 309 deaminase. It just cuts a nitrogen off the 309 to form some other amino acid, which eventually, after like 5 or 6 steps here, gets transferred into or modified into isoleucine. All right, well, you know something? Once you have enough isoleucine, you don't want to keep making isoleucine because that would be wasteful. So when there's a lot of isoleucine, we have a whole lot here, some of that isoleucine will actually bind to the 309 deaminase at this, oh, I'm sorry, it binds over here. I'm sorry, let's undo that. All right, so what happens is, now it's going to change color on me, isn't it? Some of that isoleucine is going to bind right here, okay, and then block that. So when it's bound right here, it shuts down the active site and prevents 309 from binding at the active site. So this would be an example of an allosteric inhibitor. All right, so isoleucine, the product, when there's enough of it, we have a whole lot of this right now, shuts down its own production. It shuts down 309 deaminase by binding right there. When the isoleucine gets used up and the isoleucine gets consumed and it's gone now, well, then this opens up again and it makes the active site available again for 309 to bind and do the whole thing again. So we call this negative feedback. Why did I put a negative sign and then write negative? Anyway, negative feedback because it's feeding back on itself onto the reaction negatively to shut it off. And the negative feedback is very, very common in living organisms because it allows efficiency and it allows, or basically prevents overproduction and waste of certain products. If you made too much isoleucine, more than you need, that would eventually get wasted, or it could be toxic. It could be bad for the cell or for the organism. So it's a good thing that we shut it down automatically after too much is made. All right, another example of allosteric feedback inhibition is that ATP inhibits phosphofructokinase in glycolysis. So this does not need to intimidate you. This is something we're going to learn about later when we talk about glycolysis in more detail. It's just an enzyme in glycolysis to go from glucose to pyruvate. There's a lot of steps between glucose and pyruvate. Phosphofructokinase is one of the enzymes that makes this happen. That's all. Well, in the process of glycolysis and the Krebs cycle and the electron transport chain, we make a whole lot of ATP. So if a whole lot of ATP is present in the cell, like a lot of ATP, enough ATP, what happens is the ATP feeds back upon phosphofructokinase and shuts it off. It has negative feedback right here. So ATP binding phosphofructokinase turns it off to prevent more of this from forming because we have enough. We have enough pyruvate, we have enough of the other processes in the mitochondria that make ATP. We don't need any more, so we have negative feedback right there.